I'm going to turn that on now. Welcome to the third Calumet Revisited Forum of the year. I am Michael Bose um, of the Association for the Wolf Lake Initiative. I am co-host with Dr. Valerie Penniman, Professor Emerita of History at Calumet College. Before we hear from tonight's speaker, are there any announcements? If not, I might bring you up to date on the latest at Wolf Lake. When I talk about Wolf Lake's mute swans, I mentioned that if they could speak, it would be in Chinese. That's where they come from. Tonight, we'll learn about the freshwater jellyfish, which also hail from China. Whether they speak Chinese, I believe, is mute. We might ask tonight's presenter, Dr. Nadine Folino-Rorem, an aquatic biologist at Wheaton College. She discovered the invasive jellyfish at Wolf Lake. She did so in concluding her work for the Illinois-Indiana Sea Grant. After her presentation, we will have a question and answer session. The presentation this evening is being recorded. It will be available within a week on Ollie's website. Would everyone please remain on mute? Here is Dr. Folino-Rorem.
Thanks, Michael. Thanks so much for having me join you tonight. I was telling Michael earlier, I might have a little too much information, and academics tend to get a little too excited and talk too much, but I'm very, very sensitive to your time. So I'm hoping not to do that. I may jump a few slides here and there. What I'd like to do is summarize the work that we've been doing for the Illinois-Indiana Sea Grant that we received back in 2022. And the title of our work is Investigating Environmental and Genetic Cues that May Relate to Blooms in this Invasive Freshwater Jellyfish. I just want to point out that this is a co-collaboration with Dr. Paulin Cartwright at the University of Kansas. So we've been working together on this grant along with our students. She has some graduate students, and I'm at an institution that consists primarily of undergrads. So I'll go ahead and get started and tell you about our work, and we'll get to the part of where Wolf Lake comes into the picture. You don't hear Wolf Lake right away. Trust me, I'm going to get to talking about Wolf Lake. But here's a general outline. I'll give you a little introduction to freshwater jellyfish. Most people say, freshwater jellyfish, really? And there are a few of them. The majority of them are marine. And then I'll tell you what motivated our study and the genetic objective, or the general objectives of the Illinois-Indiana Sea Grant. And then I'll get specific about the work that we did at Wolf and what are some of the implications of that research, and then time for questions. All right, so my next slide here, let me get down here. Yeah, all right. So just to make sure we're all on the same page, I wanted to give you a little bit of information about what we call the naming or the taxonomy of organisms. And some of you may be familiar with this. This may be new to others. But in the sciences, we group animals by phyla, and I'm focusing primarily on the phylum Cnidaria. So most people know that phylum, you know, if you've gone snorkeling and seen coral or sea anemones and things like that. So there's this phylum Cnidaria, and then there's a term, what we call clade or subphylum. Those are just subgroups. And those are broken down into two subgroups, the Anthozoa and the Medusazoa. And what I'm talking about, a jellyfish is really a medusa, like an upside down umbrella. And there's this group called the Medusazoa, and that includes these groups right here. And this is where we're going to be focusing tonight in this class, Hydrozoa. So this freshwater jellyfish is really a hydroid. And that gets a little confusing for some people that have had general biology because they know that jellyfish, or some people know that jellyfish are in this class down here. So those of us that are geeky about invertebrates and teeny little animals, we call the scyphozoans the true jellyfish, but hydrozoa also have a medusa stage. So these are the categories here, and I don't know if somebody needs to mute, but I'll move on to the next slide. Okay, this is just a
our friend. He was here.
A pretty picture here of Medusazoa,
Over to the beach,
and just to show you the diversity here. So the Portuguese man of war
went over the bridge.
is.
Just let me off here.
Are you all hearing what I'm hearing?
Over the bridge. And then I came back to pick him up.
We're all hearing it.
And he said,
"Okay,
hey, everybody,
from
mute."
the bridge in, it was bumper to bumper. So he's like,
somebody's on a bridge."
Okay.
Okay, how's that? Okay, I'm back.
Okay.
Yeah, great. That's probably the way to do it. So this is just a card a student gave me in the Medusazoa. And most of these, again, are the jellyfish that we all think about that are in salt water. But there are these hydrozoans that have this little jellyfish-like stage. And some of you might know the Portuguese man of war, which scares everybody when they're snorkeling. And that actually, by definition, is not really a jellyfish either, a scyphozoan. It's a hydrozoan. So just a fun fact to tell your friends and family. And here's a little bit more of that naming or taxonomy, and I don't want to overwhelm you with the science, but this is just a summary again of that subphylum of the Medusazoa, this group. And so we're gonna live right in here in this class called the Hydrozoa. And there's this body type called the polyp stage, and that's the primary dominant stage that we see in the life cycle of these organisms. So here's a little polyp stage here. It's this little stalk with tentacles. That's Hydra. It's a green Hydra. And then here's a close-up shot through a microscope of an animal that I've spent probably 30 years working on. This is another hydroid called Cordylophora. And then these down here in this class, again, these are what we call the true jellyfish. And this is Aurelia. You may see these at zoos and aquaria and when they have setups for marine displays. This is one of my students snorkeling in Belize, and she's putting her arm right next to this big jellyfish Aurelia because she worked on a project that we did for Brookfield Zoo, and she was so excited to see one of these in Belize for the very first time after she'd been working on this back at Wheaton. And then there are these cubozoans, these box jellies. They're known for giving you a pretty good sting. And then this last subphylum or class are the anthozoa, the sea anemones and the corals. So what I want to point out in a lot of the organisms in this phylum is that a lot of them are gelatinous. And so when we talk about jellyfish or blooms of jellyfish, which I'll get to shortly, oftentimes we're talking about a group of gelatinous type organisms. So again, we're going to focus in this class Hydrozoa and look at that little organism called Craspedacusta sourbi. That's the genus and species name. And it's a freshwater jellyfish in these two groups, as I said. And this is a chart that I have in my lab at where I teach at Wheaton. And I just retired actually from there. I'm still doing research, and I might be teaching a few courses down the road, like marine biology, but my load is not as heavy as it has been for 31 years. But here's this class Hydrozoa, and again, here is a Medusa stage and a Medusa stage. And again, these are not true jellyfish, though they are Medusae. So we call them Hydromedusae versus Scyphomedusae, this group down here. Just wanted to bring you up to speed on where we're hanging our hat in terms of talking about things. So here's a picture of this little beast, Craspedacusta sourbi. This is a jar of them that we found at an amazing site south of where I'm at in Wheaton, in Coal City, Illinois. And I want to show you this little video clip. I'll tell you all about the biology of these, but here they are swimming. And I'll talk more about what a bloom is, but we went to one of the bodies of water on Labor Day of 2023. Actually, it was my husband and I celebrating our anniversary on September 2nd. He's such a great field assistant. And we just happened to stop in and put the canoe in the water and went out there. And there were literally thousands of these things swimming around. So we came back and kept them in the lab for a while, and that assisted our work. So you can see that they're transparent, and we'll look at the life cycle here in a second. But they're really beautiful little things, but not so easy to spot. Oops, I'm trying to get to the next slide. Here we go. So let's take a little time to look at the life cycle. They have two phases of the life cycle. There's this asexual phase, and that just simply means that they bud little parts, like they grow like a plant. And then there's the sexual stage where the medusae, or that jellyfish-like part of the life cycle, they'll produce egg and sperm. So if we look at the mature medusa that's developed gonads or reproductive structures, they're either male or female. So they're what we call dioecious. And they'll release egg and sperm into the water. Those get fertilized, and they form this teeny little microscopic stage called a planula. That thing settles down and it'll attach to like a rock or a twig or something. And then it forms this little polyp. And the weird thing about Craspedacusta is that the majority actually of the polyp stages of hydroids have tentacles, and these don't. They have this little nubby area right here on the end. And you know that this phylum in general, they capture their prey with little stinging cells. And so people often ask me the very first thing, will these things hurt me or my family? They will not. Your skin is pretty tough. But this little hydroid doesn't have tentacles. But on that little nubby area, there are those stinging cells, and that's what that little bumpy stipulation looks like. And you can see it here and here in this picture. So this one polyp will bud another polyp, and that's again what we call kind of asexual reproduction, no egg and sperm. Here's the funky thing about this. Then the polyp will bud a medusa, and this little teeny medusa will grow up and mature and then produce egg and sperm. So this is the only sexual part of the life cycle right over here. There are a variety of other parts of the cycle where these polyps will produce these little things called frustrals. They look like little hot dogs, and they're pretty small. They're about 1/2 millimeter in size, and these little polyps are about 1 millimeter in size. So I did a little converting, and I think some folks know that there are about 2.5 centimeters in an inch. So 1 millimeter is really, really pretty small. These polyps, in addition to releasing these little hot dog things, they also can regress down into a little outer covering and form these little dormant stages called podocysts. And those are pretty small too. They're about an 1/18 or a 1/20 of a millimeter. So again, really, really small. So I think you're getting the picture by now that these things are hard to see. And the medusae, how large are they? Well, they get to be about the size of a dime or a nickel. So here are some other pictures for you to look at. And the budding polyp here, these are two polyps. These little circles are actually residue of the podocyst or the dormant stage. And once conditions are favorable, these things will regenerate and that polyp will grow out of that. And that's about 1 millimeter in height. And then here's that frustral, about 1/2 millimeter. Here's that little podocyst that has an outer covering. And then here is a medusa, about 20 millimeters, or about the size of a nickel. I was doing a little measuring today. So here's a nickel. So that's about as big as they get. They're typically about the size of a dime. They get to be about a size of a nickel. And if they're doing really well, they might get to be about the size of a quarter. But this little teeny baby here is upside down. It's approximately 1 millimeter. So again, you can imagine finding these or seeing these is a little tricky. But I'll tell you more about what we've been doing. And this is one that we actually raised in the lab. So that was pretty exciting. This simply was one that would bud off of one of these polyps. This organism, I obtained from some people down south. Some of you, if you know Mermitt Springs, which is way down in the tip of Illinois, and Mermitt Springs has it's a quarry area where they do some diving to train divers, wreck divers, and they've got some beautiful arrangements for training divers, and they see these things all the time. So we went down there one time and did some diving. We didn't see them then, but they shipped me some up. And so here are the tentacles on this little Medusa bell. This is the mouth, and then these are the gonads on these little radial arms. And this one didn't develop the fourth gonad. You know, there are variations in all kinds of organisms. So here's another schematic of the life cycle. And this is similar to what I told you before, but here's the kicker here. No one has yet to complete this life cycle in the lab. And some of you may think, well, is that a big deal? Well, for scientists, it is a big deal because a lot of us have been trying. And the people that have probably come pretty close are some scientists at some aquaria in Japan. And then we've done a little work and we've come closer, but none of us have been able to get the medusae to mature in the lab and then release the egg and sperm to get to this planula stage that takes us to the polyp. So that's of interest to us there, getting that part. And I'll say a little bit more about that, but that was one of the things that prompted us to want to write this grant and check this out. So where do we find this thing? As Michael told you, this thing originated we believe, in the upper Yangtze River Basin in China. How did it get to where it got? Well, it's proposed that maybe in the 1850s, these little teeny polyp stages were on plants, and those were perhaps transported to some of the gardens in London and England areas, and perhaps eventually things were shipped to the US. Some propose perhaps on the legs of birds. You either believe in this bird theory or you don't. We can talk more about that perhaps a little later. Or in ballast water on ships. So this is a paper that's recently been published by some colleagues showing you the distribution of where these things from the literature versus a website. It's actually the Global Biodiversity Information Facility. And you can see that these are pretty much all over the world, except for in Antarctica, in the colder areas. But notice that the majority of them are right about in this area here. And you can see that there's quite a bit of literature documenting the presence of this beast, especially in our area. And there's a lot of literature reporting the presence of this organism in Indiana and Illinois. But is it still there? That's a good question. And that's part of the work that we were looking at in terms of revisiting some of the sites. So if we look at more from this paper primarily is published to make predictions about how scientists believe that the spread or the distribution or the range actually of this organism will expand over time, especially by 2100, as water temperatures begin to increase. Many of you know, you're aware of the news that water temperatures are increasing and climate change is the issue behind that. So a lot of people are looking at a variety of organisms and how they're being affected. And we're looking at this invasive species and how aspects of its biology might be affected.
The auditor, when they're going to assess the value of the company, that the
You want to mute them all, Michael, and I'll mute back in. Unmute.
Okay, Nadine?
Okay, great. How's that? Yeah.
Thank you.
Yep. So this is from the additional information, from that paper. And a couple things I just want you to see here is that the number of recordings over time has increased. And the authors are proposing that perhaps this organism and its distribution has increased as well as the efforts to document it have. And then they've looked at records of where do we find these things, lakes, ponds, reservoirs, quarries versus rivers and canals. And ND means no data here. And these are the primary areas where you see this organism. Having studied it now for a while, you sort of get an intuitive sense of whether it might turn up where you're looking or not. And lakes, yep. Is it present in Lake Michigan? It is. In the polyp stage, have I ever seen the medusa? No. Ponds? Yep. Reservoirs? Yes. Quarries? Definitely. And then in terms of continents, you can see, as I pointed out in that previous figure, that we have quite a few data points looking at where these things, a lot of data points, recordings from North America and Europe and some in Asia. And again, some of this may be due to scientists and people looking for the beast as well. But even so, we've got people that do travel and look at some of these areas. So that gives you an idea of how common this is. And then we ask some questions of like, well, if it's so common, it's an invasive species, it's out of its range, that's what that means. So what potential impacts might this organism have?
Easy access. So now I'm with my son, all his roommates, and I said, so I've been corresponding with this guy. He's a professor at Eastern.
So if we look at this aspect of what we call jellyfish blooms, that's what really promoted us to check this work out. You've heard of algal blooms, maybe? Well, there's such a thing as jellyfish blooms. So if we look at this picture here on the upper right, look, I mean, it's just a plethora of these things. And that just refers to a high density of a bunch of individuals in a given area. And typically with jellyfish, it's a seasonality factor. These organisms increase usually when food is abundant. And a lot of these we've seen and observed in marine systems. And these are some potential causes. People are suggesting climate change.
Oh my gosh.
So when I called him back, I'm like, oh, you know what?
I'm there.
Okay, Nadine.
There we go. Okay. So increase in temperature, eutrophication, that's refers to nutrients being added into the water. So perhaps there's increases in phosphorus or nitrogen, and that can lead to zooplankton, which can lead to these things increasing in number. And also overfishing. That can tip things and lead to the increases in these as well. Because if you remove the fish, that's going to affect the food chain. Maybe if the fish aren't feeding on the plankton, the zooplankton, then these things feed on the plankton. That may lead to an increase in these. So what's the big deal? What's the problem? Well, for marine species, people don't like to swim around these if they're the kind that might sting you. They can reduce the fish populations because they can eat what the larval fish eat. And actually some of them can catch small fish. And they can clog the nets of aquaculture. Big globs and masses of these things can interfere with that process. And they can also clog power plant operations as well. Some of the power plants will intake some of the water to use as cooling. And these gelatinous globs in big numbers can really be problematic. So that's what I mean by jellyfish bloom. So the question is, does this little teeny thing, Craspedacusta medusae, what about blooms? Are there enough of these to cause a problem? Well, we don't know a lot about this organism. And that's, again, why we wrote the grant. So previous research, people think, you know, this little critter is not really a problem in freshwater systems.
I said, Kathy, will you go on the Can you go on a date with me? I said, did you hold her hand?
So, so it's not a concern, but people are wondering, could it have an ecological impact? You know, maybe play a part in the food web by eating some of the plankton that the fish eat, and maybe it can tip that a little bit. I don't know.
The balls are out of my head. I'm like, I don't like balls.
Or like the marine jellyfish, it could increase in numbers due to environmental conditions, specifically climate change or increase in water temperature. So that's really what is driving us to ask these questions, because there's quite a bit of literature and research that's documenting the increase of marine jellyfish in certain areas. So that's what led us to things. So here's my little outline from the beginning. So I've given you a little introduction on the freshwater jellyfish, the motivation for our study. So now I'll give you some of the general objectives of the grant itself and drill down into the research that we conducted in Wolf Lake and go on from there. This is a lot, so let's just sit here for a minute. But our goals were primarily to ask these four, or to address these four areas, mainly in response to, is this organism responding to climate change, perhaps, or increases in temperature? And might we see an increase in these organisms in aquatic ecosystems? And so we're just homing into bodies of water in Illinois and Indiana. So what are the environmental cues like temperature, pH, nutrients for Craspedacusta blooms? We can then use those cues to maybe predict when and where we might see an increase in these numbers in the southern Lake Michigan area. We want to replicate these conditions, meaning these factors that we see in the field, and try to do that in the lab, grow them in the lab, so that we could get medusa formation in the lab and then try to keep them to reproduction, since no one's been able to do that for very long. And then we want to look at genetic mechanisms, like, do we see any similarities or differences or aspects about genetics that might be involved in this organism's ability to do what it's doing in new habitats, and how that might relate to or parallel what we see with marine jellyfish. And then our last goal was to improve scientific literacy and outreach. And that's part of the reason why, like social science, part of the reason why I reached out to Mike, and I'm really excited to be able to talk about this, because the more people know about this organism, the more communication we can have, and there's greater potential for gaining information about location and different sites and things like that. So let's look at the first goal. The field and laboratory part. These are some things we looked at in the field. The parameters that might lead to the blooms. So we'd go out and we looked at temperature, pH, phosphorus, calcium, nitrates, whether that be ammonia and such, conductivity, or the amount of salts in the water. And then the Secchi disc is a device that you look at how turbid versus how clear or less clear the water might be. And we went out in the field and we found 3 positive sites in Illinois. And then we found 2 positive sites in Indiana, one of them being Wolf Lake, and should have put the other one in there. The other one is, it's a marina nearby, just across the border. Sorry, the name blanks, I blank on the name right now. But we also did some replicate works in these artificial ponds at the University of Kansas to see if we could see how these parameters might measure to what we were seeing in the field. This is Dr. Cartwright bending down. This is the outdoor lab tech. And then this is Kent Wanata, her PhD or master's student actually at the University of Kansas. And then I had students help me. We'd go out in some boats, and these are 3 students here, Chloe Davis, Jensen Langseth, and Micah Perry. Jensen is a senior now. She's still working with me this year. So that's been kind of neat. So here's a little bit about culturing them in the lab. We are able to culture them in that little teeny polyp stage, and we are able to get them to produce medusae. This is the setup at Kansas. in terms of what they grow. They would grow them in these dishes and petri dishes. And I wonder why that's all coming in on the queue there. They must have set that slide up a little differently. I think that's everything. There we go. Okay. So we would get jellyfish and keep them in small containers and cover them. And this is a little instant ocean. So a lot of us would make up really, really dilute solutions for these things since they're fresh water. But they'd keep the jellyfish in some of these taller beakers and then petri dishes for some of the polyp stages. And some of you may know sea monkeys are really fun to hatch. That's why you have to have salt water. They hatch in salt water. And we'd feed the like a 1-day-old sea monkey to these things 3 times a week. And we culture these in what's called hydromedium. Heard all about Sharon. And it's called HM for short. And that's a special solution that somebody developed a while ago. And a student and I, 10 years ago now, 9 years ago, figured that hydromedium is a really great solution for Craspedacusta to grow in. So that's become sort of a standard now to try to culture these in the lab. Here's a little bit of Wheaton. This is Micah here and Jensen, and they are looking at medusae in some beakers that we have here. And these are our jugs of HM. We'd make really large amounts, and we would culture our polyps and things in these little pudding bowls, and we would keep them in this controlled temperature chamber. So that was pretty nice for the setup that we had at Wheaton. So related to that goal of replicating these conditions in the lab so that we could medusae, we did monitor these factors of pH and flow and warmer water. That tended to work better for a lot of what we were doing. And then we tried to start feeding them things besides the brine shrimp. Here's the tricky part. And what are you seeing here? These are little containers like what you buy your mixed nuts in at Costco. And the engineer students at Wheaton got really excited about this work. And so they 3D printed some of these paddles to create circulation for this, for our work. So we got little motors on top right up here. And it was really neat to get the engineers involved. And here are 2 little medusae that we've cultured from budding off of the polyps. And they're orange because that's how the shrimp hatch out. But the shrimp hatch in salt water. And we rinse them 3 times, but I still think that we really need to work on getting freshwater food for them, copepods and cladocerans, these little zooplankton. So we've been trying to culture some freshwater algae to then feed the copepods and the cladocerans to then feed the medusae. But it seems like we get one going and then we don't get the other one going. So we haven't been able to get that worked out yet. But that's just another part of trying to replicate the field conditions in the lab to see if we can complete that life cycle. The third goal relates to the genetic aspects of things. And those genetic cues, if there are any, were looked at closely by Dr. Cartwright's student, Kent, and that was his master's dissertation. And we looked at a couple different populations. We had a Japan strain and a Panama strain that served somewhat as like a baseline because we had worked with those in the past. And this is an Inverness, a strain from Illinois, and then another strain from Lawrence, Kansas. And this simply shows you the average number of those little polyps over time and at 4 different temperatures. And these polyps form from the frustrals. So we started with the same number of frustrals roughly, or standardized that, put them in a bowl, and then we'd look at how those frustrals would turn into polyps and then how those polyps would multiply in number over time. And so we didn't observe any major differences, but the genetic variability that Kent was able to observe shows us that these organisms can survive in a wide range. So we've got different populations with incredible physiological ranges, if you will, in terms of temperature. And even though they're from different places, they all seem to grow well, or if we say optimally, around 26 degrees C. So that's fascinating when you think about an invasive species. If you're an organism that's expanding into new habitats, your ability to acclimate physiologically and set up shop and get established is pretty important. And so this organism has obviously done very well. And that's one of the attributes that we see when we think about invasive species. They can be introduced to an area, but then can they actually become established and reproduce? And this little freshwater jellyfish seems to be doing that. All right. So here's where I want to come back to this first goal of ours. So we talked a little bit about the environmental cues and the conditions in the lab and the genetics. And I want to take a little bit of a detour because this is where Wolf Lake comes in. You're probably thinking, finally. So you're probably wondering, how did we know where to find these? I can tell you I jumped on a lot of bodies of water. We have some previous information of where these have been found in the past. But here's the kicker. The medusae do not show up or are not seen consistently at locations. You may see them for 5 or 6 years, and then you may not see them for a while. So the question is that because people aren't looking? Is it because they're not there? Is that because there's something affecting some aspect of that life cycle? So typically what we do for sampling is we go out in the field and we pick up rocks and twigs and things. And that takes a lot of time, We spend a lot of time. So this project that I'm telling you about, and part of it occurred in Wolf Lake, was thought up by an undergrad at Wheaton, Jonathan Zhu. Jonathan's now finishing his master's at Carnegie Mellon. He's a really, one of those quiet, bright students and really unassuming. And he came into the jellyfish lab and joined the crowd and worked on it for 2 years. And now he's still continuing to work on that. And it's been a real joy to work with him, but it's been neat to see how he's grown as a scientist. So he had this idea of how can we locate and sample in a way that's going to save us time. I mean, we're talking hours and hours. So he thought up a new way of sampling for these things in aquatic habitats. And here he is looking at some samples here. These are temperature chambers. So I'll give you a little bit of history. So you history buffs, how do we typically sample for this organism? Well, we look for the medusae. There are more articles reported on the medusae, people seeing the medusae, if they do see it, than they are on these teeny little stages of the polyp or the frustral. And here's a little colony here. How do you collect the medusae? You take a net or a jar and you scoop it up out of the water, the little medusa. And again, the polyp and the colonies aren't sampled as often because they're so small. But it's really, really important to find these things because if they're present, then you should see the medusa appearances. But those are sporadic. So here's the question. Do we see the polyp all the time, but we don't see the medusa because there's something happening in that life cycle component that's keeping the medusae from being present? Or are there lots of medusae and people aren't out there? Or are things eating the medusae? Some people say that fish do not eat them, but I have spoken to fisher people, and they do eat them. So there's no systematic method to sample for this polyp stage, other than you go out and get rocks and sticks and plants, and then you bring these rocks and sticks and plants, and you're trying to manipulate them under a microscope. And I can tell you, we've spent hours and hours and hours combing every surface of some rocks and pieces of wood that have fallen into the water and looking at them under a microscope. And we're looking for things that are 0.5 millimeter or 1 millimeter in size. And when they're not fed brine shrimp to turn them orange, like you see here, they're very transparent. So Jonathan came up with this idea, and it began with some preliminary lab work. He read in the literature that people would find these polyps on plastic and glass. So he was wondering which of those might be better. So he used a method that I use with another hydroid that I mentioned that I've been working on for a long time of growing them on slides. We usually cut the bottom and the top out of this slide box. It's a few inches by maybe 2.5 by 4 inches. And then we got plastic slides and glass slides. He took the little frustrals, those little hot dog things, and he put them on there, kept them horizontal, and polyps attached. So the frustrals turned into or morphed into the polyps and they attached. So he was able to, you can see these little things on here, these little specks, those are all polyps. So he was able to culture those in the lab. So then he thinks, all right, let's go to the field. So plastic slides, glass slides, and then these glass petri dishes in those little cages that you put your woodpecker suet out. And then a common device that's called a hesterdendi. These are like compressed wood that the EPA uses. It's a disc about that big, standardized area. So we put these together, and you can see we put them together, put some floats on them, and clipped them to a rock. And here's Micah getting ready to deploy one of them. And we put them at 3 sites. And we left them out for 2 months to see if we would get polyps or frustrals on these devices at these 3 locations. 3 locations, finally, Wolf Lake. So we had a location in Illinois, in Coal City, and at Wolf Lake. And we had 2 complete setups at each location. Well, we had 2 complete setups at 2 locations. We had, let's see, 2 complete setups at 2, yes. So I put 2 setups here, and I put 2 setups here. So 2 were behind Boy Scout Island, and 2 were in front of Boy Scout Island. And unfortunately, when we went back out to retrieve these, the ones on the front had been vandalized, meaning the markers or the little buoys that we had put on them had either been cut or the devices had been pulled out completely. So our sample size went down to half, which was kind of a bummer, but that's okay. We still were able to analyze the data. And this is what we did. We brought these, all of these back to the lab, and then we kept them in the lake water, and we looked at it. We looked for polyps, we looked for those teeny little podocysts, and then we looked for other invertebrates, and we counted those. Here's a dish with one of the hesterdendi plates in it. Here's a cooler. We put everything in an individual Tupperware container and brought that back to the lab. That took a little bit of time as well. Here are some results that Jonathan found. And the cool thing is that Jonathan presented this work at the International Invasive Species Conference last May that was held in Halifax, Nova Scotia. So that was pretty exciting, and published a paper as well that came out after getting feedback from people. What did he find? What did we see? That the glass slides are most effective. So here are your glass slides, and he took our raw data and converted it to the number of polyps per square inch. And we modified that and had to change that to metric when in the final paper, but this is one of his preliminary slides. We didn't see things on the hesterdendis at all. We didn't see things in the petri dish in terms of polyps, and we saw them on the plastic slides. Okay, and he found some significant differences. And he found more polyps at the Barrington site than some of the other sites. Okay, and these are the frustrals that we found, those little teeny hot dogs, again, glass slides and plastic slides. So we've got some ideas about these, why we didn't find them, and I'll mention that in a second. So the invertebrates that we found are zebra mussels, oligochaetes or worms, freshwater like friends of earthworms, these chironomids or larval stages of insects, and then hydra, another hydrozoan. And we found these things, quite a few of these, more of them on the hesterdendis, and we saw some on the glass slides and the plastic slides in the petri dish. Now, the hesterdendi, again, is a device that EPA uses to assess macroinvertebrates to then use as indicators for water quality. So not surprising that we didn't find that they found so much on the hesterdendis, the other invertebrates. But again, we're kind of puzzled, still trying to figure out why we didn't find the podocysts or the polyps on the hesterdendis and the petri dishes. We did find the podocyst at all the sites on the glass and the plastic slides. And there's that podocyst again. Okay, so some ideas of why. Well, let me go back for a second right here. I believe that there's so many other invertebrates, and these chironomids, they are really voracious little insect larvae, and they will eat the polyp stage. And I've seen them do that in the lab. So I'm wondering if we did this experiment for a shorter period of time, which is what I'll mention on the next slide. And the Petri dish might be the same, is that we might have organisms here that may be eating the stages of the Craspedacusta before we get to it. So we're going to do, and it may be the positioning too of where we had these things. I'll move on to the next slide about that. These really allow us to confirm the polyp stage, and we might be able to obtain specific populations of Craspedacusta at different sites. Positional changes, what I mean, well, we had certain ones always down by the brick and then the ones at the top. So the Hestrodindes were up top. And so we did an experiment where we switched them, and our preliminary results still show us the same, that we're not getting them on the Hestrodindes. That's why I think that something's eating them, or we need to deploy them for a shorter length of time. And there may be some seasonality aspects. So, but here's just a picture of, again, why it's important to find the polyp stage. If you don't see the medusae, or this is a picture at Cold City of medusae in the water, it's really hard to see them until the temperatures really spike, and then you can really see them pretty well. And here's what those little polyps look like from the field. Okay, so further studies, we might try other substrate types like stones or plants or pieces of wood, or even zebra mussel shells. And there are areas without polyp colonies reported or collected, like Mermit Springs. We could put these in there and see if we get polyps. We should get polyps because that's where they see a lot of medusae. And there also may be some aspects of flow, because these little frustrals were loose. It's kind of like a spider web balloon or a spider ballooning. They produce these little mucus threads so they can disperse around. And this is Hydra eating a frustral. So that again comes back to the predator part of things. This is a picture of one of the substrate types. This looks like the Petri dish where you have Hydra and some bryozoa and a bunch of other things. So easily those frustrals could have been eaten by either Hydra or some of the insect larvae. So there's predation by chironomids, or even competition for space from these bryozoas. So they can be some other factors there. So our last goal is the scientific literacy and education. And just to summarize what we've done, and our goal is to increase awareness about invasive species. So we developed some lesson plans, and this was a student, Sarah Chang. She now teaches at the field school in Chicago. She developed some plans, and actually the field school students came to Wheaton, and we taught them about invasive species and about this jellyfish. Dr. Cartwright has presented to several school groups that have come to her lab, and we've published some of this work. We're trying to get some relationships going with iNaturalists, and we're hoping from, we might still have a school or two come to the lab, but we've wrapped things up. But we really would love to do more outreach to school groups if we can, but the funding is complete now, so we might need to reapply for that funding. But this is where we are, Wheaton. This is a lab, and we brought Dr. Cartwright in via Zoom since she was in Kansas. So these are all the students from the field school, mainly middle school age. And I'm explaining the life cycle. And then we gave them each their own bowl to take a look at this. So it was really pretty fun. So the last, wrapping it up, implications of this research. Well, our research results hopefully will be helpful. We've published a couple of papers from this work, and hopefully it'll shed some insight on what are the ecological impacts of this jellyfish. When do these blooms, or are we going to see more blooms? We know more about what cues might be more important. Temperature seems to be the primary cue, but you need to have the right habitat. We'd love to be able to complete more of the life cycle in the lab to better understand those cues. So we're still working on that, and I still would love to get freshwater food cultured in the lab to feed them. And then we could also gather information about invasive species when they are in new environments. Are there genetic aspects or variation within a given species that enhances its ability to become established? And that might help us make predictions. And lastly, as Jonathan's ideas came to be, we did develop a sampling technique that might be applicable to other people. I know some people at the conference were saying, you ought to market these things. So he did a really good job explaining to folks that this is a really plausible method that might be helpful. Okay, here's some contact information. If you see these things or have questions later or want to email me, please feel free to reach out. Or if you know people that spend a lot of time on Wolf Lake, maybe fishing, and love to kind of get them to maybe look for these things. And just some acknowledgments to the college and their support and others that helped us put these things out in the field. And again, the funding from Sea Grant, the Illinois-Indiana Sea Grant. And here is our infamous dog, Pepper. If you have any questions, I would love to entertain those questions. Thank you.
Thank you.
Yep.
Are there native freshwater jellyfish?
There are not native that I know of. There are only 3 that I'm aware of, and globally, and the other 2 are not in this area as best as we know.
What is the evidence that these freshwater jellyfish all over the world came from a single location in China?
Good question. There have been several people that have done some molecular work, and we look at certain enzymes that change over time, and we can use those to give us the capability to map and look at the distribution pattern. So that information, along with the presence or the distribution where people find them, suggests that that's the origination.
Excuse me, Nadine. How are you doing?
Fine. How are you?
Okay. I'd like to find out if the jellyfish is a threat to our fishes in our area if brought here.
Well, it's here. It's in Wolf Lake. The question is whether it would become more abundant. Most people think that the numbers are fairly low in terms of the impact they would have on the food that the fish feed on, at least the larval fish and the juvenile fish. But the question that many, many people are asking in both marine and freshwater systems, if we get these blooms or these moments in time where there are so many of them that they might eat enough food that they diminish the food that's available to the larval fish, then that could be problematic. But that's always the question that comes along with the temperature increases. So
How can the jellyfish be controlled? What methods should we use to keep the fish to keep the jellies in control to save our native aquatic life?
Oh, good question. Well, there are a couple of ways. If they were to become a problem, I think what some folks have potentially done in marine system is that in given locations or habitats, you can strain these out. But for example, there's this other gelatinous organism called a ctenophore that actually went from the United States over to, I think, Europe, and actually caused a crash in the sardine population that was supporting people in that area, I think near Turkey. And that was very problematic. And there wasn't a lot that they could do. So that's again why, if we understand aspects of the life cycle, there might be some decisions that could be made that would target the organism without harming the organisms that typically live there. You don't want to just want to dump something in there. I mean, you're going to harm other things as well. So maybe mechanical removal or things like that might be the best way. But on a scale, I mean, that's huge. But again, it depends on the locale and the abundance of the predator. Right now, they don't seem to be a problem, but we're doing this preliminary work in case we might see more of them as temperatures increase. And it's true that the water temperatures are increasing in Lake Michigan and surrounding areas. We've got some long-term temperature monitors out, and we haven't made comparisons to last year, but I don't know if any of you are gardeners. I'm a big gardener, and I'm kind of chewing my nails a little bit. I do a lot of winter sowing in milk jugs, and I have prairie plants that need to stratify, and these temperatures just aren't staying cold. So we'll see. I'm gonna have to put more things in the fridge because things are just warmer than they typically are.
And are jellyfish a danger to humans?
Not this species. Marine ones can be harmful, but this species will not be harmful for you in terms of stinging you or anything at all. They're too small and your skin's pretty tough. It startles people when they see them, but they're really pretty, actually.
Could they spread as far as into Lake Michigan if they're not in check?
I missed the last part. They are in Lake Michigan.
And how dangerous are to the native aquatic life over there if they're not found and kept under control?
They're not dangerous at this point, again, because of the abundance being low and the size being small. Yeah.
Nadine, there's a couple questions
Yeah.
On chat
Okay. Let's
that
see.
they may have already been answered, but let me just read them to you.
Great.
Just to clarify, what are some of the possible ecological impacts of freshwater jellyfish blooms?
So if the jellyfish, if there are so many of them, they get so abundant, they can eat food that other organisms need to eat, like the fish. And again, we've seen these ctenophores do this with the sardine industry outside of Turkey. But you would be talking about localized areas. Lake Michigan's a pretty large lake, right? But there could be some localization where the jellyfish would eat the food of the other organisms. And the polyp, they may eat some larval stages of insects, which again, not just zooplankton, but maybe some of these smaller insect larvae that are also important. They're aquatic, and those are important for fish as well. So there's a lot about that, their feeding biology that we don't fully know. Whether the jellyfish can actually capture small fish and eat them, we don't know that either. So,
Has anyone seen Medusa in Wolf Lake?
not that I'm aware of. That's why I would love to see. I know quite a few people fish there, right? And when I was there doing this work, I know the visibility of the water is fairly low. And I did do some plankton tows, seeing maybe I might see some medusae, or catch some medusae in my nets, and I did not. So there's no record of it in Wolf Lake, and I happened to find it just by my first visit there. I picked up some branches along the shore, waded in, and took them back to the lab, and sure enough, the teeny weeny polyp stage was on there. So that was a kind of a random find, if you will. I'd like to visit and maybe do more at Wolf Lake if time or funds would permit, but we're sort of at a transition phase here. But I'm curious, very, very curious.
Is it typical that you would find individuals like that without the bloom?
Yeah, you could find them. But it seems to be very seasonal in temperature. Like when we saw that massive amount at Coal City, that was September, what was it, the 4th or 5th, and the water temperature was almost 80 degrees because they have small ponds there. And the bloom lasted about 2 days. And we went back about 4 days later, we could barely find any. So did they swim down? Did the fish eat them? I don't know where they all went. It was really fascinating. And that's why I wish I could find a site that I could have access to that I could monitor daily during that season in the fall. We typically see these things in the late fall when the food is abundant, the water is warm. It's before we start seeing temperatures decline. So August, September, October. So maybe into July, but usually it's September, October.
And what do you say about
Oh,
the disturbed versus undisturbed areas? I know we have some like more sensitive swales near Wolf Lake in Eggers Grove there. And do you know if they would be present there or what the likelihood is?
That's a good question. I don't know. They tend to be present where there's kind of slower water movement, if you will. But then there also needs to be food availability. So it might be interesting to take a peek and poke around in some of those areas that are kind of out pockets, if you will, or little side pockets of the lake. Yeah. We were out around the island, so there's obviously quite a bit of water activity from the wind. We were out there when it was pretty windy one time.
I've got a question about your research technique. You had quite an innovative way to collect your samples using the plastic slides and also the glass slides. But you also had petri dishes in there. You didn't indicate if they were glass or plastic, and wouldn't it have been appropriate to use both? The petri dish has a larger surface area for collecting. I'm just curious.
That's a great question, Michael. That petri dish methodology, we were given that idea by Dr. Tim Wood at Wright State. Tim and I go way back. He's a bryozoan expert. So he suggested that, and those are glass actually. But you're spot on. If we were to find things in those petri dishes, we would go with glass and plastic for sure. We haven't found any aspect of Craspedacusta's life cycle in those petri dishes. And that, again, may be to the way we had them positioned. We had them horizontal. They were up higher on the brick from the bottom. So putting these near the bottom or near a source, you sort of have to know where you might have polyps that are going to produce frustrals. So they need to be near the bottom. And so we want to put more bricks out with those petri dishes closer to the bottom. And we have a site that I have access. That's actually in Inverness. It's Phyllis is her name. She's in her mid-80s and lets me take her boat out anytime. And she saw the jellyfish there over 12 years ago. But the polyp and the frustral stage are there. So we want to do a little bit more with these very things. So that's a great idea of trying to do the plastic and the glass. But we'll put those out for sure at different positions and see if we get anything. It may be a flow factor, but maybe that cage hinders things. I'm not sure.
Thank you.
I was surprised that we didn't see them in that. The Hestrodendy kind of makes sense to me, but I was very, very surprised that we didn't see them in the petri dishes. Other questions or comments? Or
there's a question from how might road salt salinity fluctuations affect blooms?
Yes, good point. Because that's a big thing now. The other hydroid I've studied for a long time, Cordylophora, would love that salt input because that hydroid lives in freshwater and brackish water both. And it tends to thrive or do a little better when a little salt's added. But from what we've seen in the different water media that we've used for Craspedacusta, that hydromedium, input of salt would probably be problematic and harmful, I'm guessing. They seem to be a little more sensitive to changes in salt compared to that other hydroid. So that would be my guess, and that would be a really good experiment to do. We did look at little bits of salinity, but not salinity straight on when we were looking at lake water versus hydromedium. And hydromedium has salts or solutes in it, but not high like one would see that we see when we have runoff from road salts. So that could be harmful. Absolutely. Not just to the Cnidaria, but to many of our aquatic invertebrates that are important for the food webs there.
Another question is, will the polyps or medusa affect frog or fish eggs?
They I've read where they can eat fish eggs, yes. Frog eggs might be a little larger, but there are a few studies that have said have documented them consuming eggs of fish. So yeah, both stages. Yeah. The medusae feed in a really interesting way. It's called parachute feeding. They sort of swim up and then they float down. And when they float down, they often will catch things with those tentacles. They sometimes will float upside down too. And then they'll often lie on the bottom. So they're going to catch things in the water column and things on the bottom as well. But the polyp is attached.
Hey, Nadine, I just want to ask another question about jellyfish breeding. How many eggs do these jellyfish produce, and how long do they live out in the wild?
We have no idea. I'm guessing because they're aquatic, they probably produce hundreds of eggs. That's typical for a lot of aquatic organisms because the probability of survival is really, really low. But no one has seen egg production in any of these. They haven't looked at it, haven't been able to assess it in the field or in the lab. So I don't really have an answer for you. Yeah.
And how long do they live in captivity?
That's been the tricky part. We've been able to keep the medusa alive for 78 days, and that I think passed the Japanese record. But after that, none of us have been able to keep them. They do this thing like what's called inversion. It's like an umbrella blowing inside out in the wind. So they lose their shape and they flop inside out, and that affects their ability to feed, and they eventually die. So it's what we call inverted. And there are records of this back in the early 1900s as well, and we've seen it in the lab. I thought maybe they need to swim, keep them moving. That's why we tried to move them. No one really knows. It might be temperature stress. We haven't figured that one out either.
Do these jellyfish adapt to colder weather, especially in the water?
I think they can survive through the winter because I know down at Mermitt Springs, the place where they dive all year, they've seen jellyfish throughout the entire year. They're not as abundant in the colder months as they are in late summer, early fall, but they said they've seen them. So again, there's a place I'd like to spend some time diving throughout the year and making observations. Other questions? If you could spread the word for folks that hang around the lake, and if they see one of these things, give a holler, boy, we'd be really curious. I'm hoping to bring the boat or the canoe back over this coming summer and see what we can do. Whether we deploy more or not, I don't know. I have to renew my permits that I got from the state, and maybe we'll put some things out for a shorter period of time, but we need to see where our funding, get some more funding for that kind of work. So, but yeah, it's kind of a fun place to find them just on a whim. No records have indicated that in the past. So.
I found the medusa in Schiller Pond in the Cook County Forest Preserve 30 years ago. I went back a couple times and never found them again. I was just, one visit, like they were there, and at least a couple other times I went there, they weren't there.
You're describing exactly the dilemma that we have, is that we see them and sometimes we don't. And so which pond was that, Dennis?
Schiller Pond in Schiller Woods Forest Preserve. It's up in the northwest area. Up on Irving Park Road, I think, in the Des Plaines River.
Oh, sure. Yeah. Yeah, the polyp stage is in the Des Plaines River. I found that because I've worked in that river a lot. Great. I just make a note of that. Okay, super. Thank you.
Thank you
Great.
very much.
Thank you all. I really
Okay.
appreciate it. And
Nadine, thank you very much. I'm sorry for the interruptions. And
no problem.
I was just reading instructions on how to solve the problem. So
All right, for your
the
next time.
next time that happens to us, maybe I'll figure it out.
No
But-
problem at all. Thanks for
but-
having me. And feel free to email or whatever in the
Yeah.
future.
Thank you, Nadine.
Thank you
Valerie
all.
and I are really grateful for your presentations, and we want to thank everybody for attending the meeting.
Yes.
It's interesting. We started out with something in the mid 30s, and then we ended up in the mid 20s. So
Yes.
so
Okay.
that is pretty good.
Okay, great. Good.
So anyway,
Well, thank you again.
Thank you for coming.
Yep. Thank you. Good night.
Good night.
Adios.
Adios.